Hello, I am Professor Sims, and this video is about hand washing, particle transmission, and epidemiology. This is the first of 10 lab sessions included in my Laboratory for Fundamentals of Microbiology course. If you're currently enrolled in this course, please consult the syllabus and Moodle site for assignments and any other course information. The learning objectives for this unit include testing the efficacy of hand washing as a control measure for pathogen transmission, performing experiments illustrating microbial transmission via direct contact, modeling the scientific method, collecting and interpreting scientific data, and understanding safety and disposal procedures relating to this lab. To begin, it is important to understand the difference between normal flora and transient flora. Normal flora are microorganisms that normally live with you. Humans have, by some estimates, 10 times more bacterial cells than human cells in their body. Humans cannot survive without their normal flora, which are permanent residents of the body. They're usually not pathogenic, meaning they do not normally cause disease. However, normal flora can become pathogenic if they go somewhere they're not supposed to be. For example, Staphylococcus aureus is normal flora of the upper respiratory tract, but if it gets into a skin laceration and enters your bloodstream, then all of a sudden you have a staph infection. Normal flora compete with and they antagonize non-indigenous species. This is good because they can crowd out and kick the butts of pathogens that are trying to make you sick, so that's good. They live in your sweat and oil glands and your dermis and your eyes and your nose and your mouth, your throat, even your urethra, your intestinal tract. Normal flora are essential. They're essential for your intestinal health. You cannot live without them. You need them for intestinal health, digestion, production of some vitamins like vitamin K and B12. Some normal flora can also produce their own antibiotics, which kill non-indigenous species, pathogenic species. Transient flora are different than normal flora. These guys are temporary residents of the human body. These are microorganisms that are transmitted from the environment or from another host. So unlike normal flora of the skin, which reside in the dermal layer, in the skin, your transient flora live on the outer epidermal layer. Because the epidermis is quite dry and acidic, it's difficult for transient flora to reproduce and survive there. So the transient flora are going to usually die off or get washed away, especially when good hand washing technique is employed. So we're going to learn some more about that in a little bit. Transient flora may be, but are not always pathogenic. Skin is the largest organ of your body, and as long as it's intact, meaning with no lacerations or open wounds, it is extremely effective as a barrier to protect against infection. Regardless of whether or not a microbe is normal flora, transient flora, whether it's pathogenic or non-pathogenic, the skin can prevent entry of microbes into the bloodstream where it can be detrimental to your health. Hand washing is the most effective way to prevent the spread of nosocomial infections. These are acquired in hospitals or in healthcare facilities. Nosocomial infections, they affect nearly 2 million patients and cause 20,000 deaths each year. For example, in 2011, more than 720,000 hospital-acquired infections occurred in the United States, according to the CDC. About 22% of those occurred at a surgical site. Cases of pneumonia were another 22%. Urinary tract infections accounted for an additional 13%. And then primary bloodstream infections was 10%, just in that year. The most frequent cause of nosocomial infection is Staphylococcus aureus, a normal flora of the upper respiratory tract. It's been estimated that approximately 30% of the human population carries Staph aureus, which that sounds like a lot until you compare that to 75% of hospital and healthcare workers. The really tricky thing about Staph aureus is that not only is it ubiquitous, which means it's everywhere, but it also can survive on the skin for several weeks. before becoming pathogenic or displaying any symptoms of infection. So I want to talk about proper hand washing. This figure here shows where most people tend to miss when they are washing their hands. It is extremely important to learn the proper hand washing technique, and even good hand washers can miss in between their fingers and in the little cracks and crevices in their palms here. The correct hand washing technique will be demonstrated in class, but it involves rinsing your hands first, then using antimicrobial detergent. The one we're using is going to be Hibiclens or Hibiclens. And then you lather the entirety of each hand, paying special attention to all of these little spots where most people miss. So the back of the hand, between the fingers, under your fingernails, up above your wrists, inside all of those little creases in your palm. And you want to do that lathering, just that part, for approximately 20 seconds. And if you're not sure how to estimate 20 seconds, you can sing the happy birthday song twice. That's approximately 20 seconds. And then finally you rinse the hands again, and you want to dry with paper towels. Paper towels are cleaner than the air blow dryers because those are actually blowing more bacteria onto your hands. For experiment 1, we're actually going to test the efficacy of your hand washing technique. So the proper technique will be demonstrated to you using a swab culture. You're going to swab your hands before you wash them and put that on a nutrient agar plate. Then you'll swab them again after you wash them and put that on a nutrient agar plate. And we will incubate that plate and we'll see what happens. We'll see what kind of bacteria are still there after you washed your hands, if they're the same bacteria that was there before, if it's something different, it's going to be fun. For a pathogen to persist, okay, in other words, for a pathogen to keep itself alive, it must put itself in a position to be transmitted to a new host. Mechanisms of particulate, i.e. microbial transmission, what this is modes of transmission, okay? These include direct contact, indirect contact, droplet contact, airborne transmission, fecal-oral transmission, and vector-borne transmission. Direct contact transmission is pretty self-explanatory. It's from person to person, from host to host. Indirect contact transmission usually involves a contaminated object, also known as a fomite. A fomite is an inanimate object that potentially transmits pathogens. That is indirect contact. Fomites facilitate the indirect transmission when they come into contact with a particulate, subsequently come into contact with the host. Okay. Droplet contact generally refers to pathogen transmission via coughing, sneezing, breathing too close to someone, all of that kind of stuff. It's been estimated that a single sneeze can send thousands of virus particles into the air. And they send it pretty far too. But for something to be considered airborne, it needs to be transmittable in the air further than 1 meter, so that's about 3 feet. So this is not the same as somebody coughing, like coughing in your face. It has to be able to travel over a distance and survive over that distance for some time in order for it to be considered airborne. Fecal-oral transmission is just as gross as it sounds. These are the waterborne pathogens that are shed in feces, and then they're ingested by a contaminated water supply. There's a very good video that I put in the description of this video explaining how cholera is transmitted and how it's an example of fecal-oral transmission. And then, of course, you have vector-borne transmission. So these are animals that are transmitting disease from one host to another. The easiest example of this would be malaria. It's transmitted via mosquitoes. Pathogens that rely on insect vectors for transmission, this is usually blood that's being extracted by a biting insect. So it's not just mosquitoes, it can be ticks, fleas, any of those kinds of things. So for experiment 2, we're going to illustrate microbial transmission via direct and indirect contact. For this, we're going to be using glow germ powder. Glow germ is a synthetic, it's basically plastic that has dye in it that glows under UV. It's used to simulate microbial transmission because the individual little pieces of powder are the size of individual cells of Saccharomyces cerevisiae, which is a yeast. But there are no toxins in Glow Germ. You can handle this stuff with your bare hands, no problem. And what we will do is we'll use the Glow Germ to see not only like how easily things are transmitted. So you put some on your hands and you touch your desk and you can see how much of that stuff actually gets transferred. And then you can also use it to test to see how well you're washing your hands. So again, I'm kind of beating it in your head. I need to make sure that everybody is using proper hand washing technique. And this is a really, really fun experiment, a really, really fun way to see that.
And you may very well be surprised by this result. Have a look here at this quick Glow Germ demonstration.
As you can see, the germs glow blue under ultraviolet light. Now what happens if I touch something like this coffee mug? Some of the glow germs get transferred from my hand to the mug. And this is just how real germs spread every time you touch something. Now I've just washed my hands. So we'll turn the light off again and see how well I did. Look at that. I missed whole patches of germs.
Finally, experiment 3 is going to be looking into epidemiology. Epidemiology is the study of occurrence, transmission, frequency, distribution, prevention, and control of disease in human populations. Epidemiologists are essentially disease detectives whose major concern is developing methods to prevent pathogen transmission. Some epidemiological terms to know are endemic, epidemic, pandemic, right? And so not only do you want to know these terms, but you want to know the difference and similarities between them. So your endemic level is the baseline, right? This is the level at which a pathogen is prevalent in a population or a geographic area at regular, normal times, okay? An epidemic is when a pathogen increases suddenly, and it's a factor of time. A pandemic is when you have an epidemic, right? So you have a pathogen that is increased in a short time, but it's not just a factor of time, it's mostly a factor of space. A pandemic is an epidemic that's occurring over a large geographic area. It's also important to understand the mechanisms by which pathogens enter the body, especially for when you're doing your lab, so you can consider these things for safety reasons. So you have lacerations, scrapes, rashes, dry skin, hair follicles, sweat glands. We've already talked about skin as being a protective barrier. You can have bacteria that not only enter your lacerations, but also pores and sweat glands. You probably have heard of acne, right? So this is when bacteria have been trapped in your pores. But you also have things like hookworm. Hookworm can penetrate the skin, so in that case, the barrier kind of breaks down there. You have exposure via breathing in airborne pathogens. So you've got the respiratory system, which is a mechanism through which pathogens enter. There's airborne transmission, also your droplet transmission, that kind of thing. You do kind of have a backup system. Your body produces mucus, which serves the very practical function of trapping pathogens, especially in your lungs and upper respiratory tract. Also, you can ingest contaminated food or drinking water, and also touching the mouth, biting your nails, chewing on pencils. You generally want to avoid putting things in your mouth. Don't put your fingers in your hands and stuff in your mouth. For experiment 3, all you're going to need as far as materials is a computer or other internet-connected device. What you're going to do is observe and interpret scientific data in order to determine the source of an epidemic. So you'll complete an online simulation provided by the Centers of Disease Control and Prevention. The case study is called Midterm Revenge, and it's located here at this website. You'll read through all of the clues, being sure to take note of them all, especially including the attack rates and control data that's included in clue number 4. That's going to be important, especially for completing your lab report template.
Make sure you go through these safety guidelines. Just a couple things I'm going to make sure I point out here. You will be wearing your gloves and glasses, so make sure you have those. Make sure you have your lab coat. If you have any accidents or spills while you're in lab, obviously you want to make sure that you notify your instructor immediately. Any kind of swabs or anything that needs to be disposed of. Make sure that you know where those go. And if you don't know, ask your instructor. Leave anything that needs to be incubated. Make sure it's labeled properly with your section and table, and leave those at the end of the bench at the end of class, and your instructor will handle those.
For your observations and interpretations, for the first experiment, you want to make note of the abundance and or diversity of the microbial growth before and after hand washing. How many different colony types are present? Was your hypothesis supported or rejected? For experiment 2, you're going to be looking at your hands after glow germ and what areas did you miss? How could you improve your hand washing technique? How did the particle transmission compare to what you expected to happen? And for experiment 3, were you able to successfully trace back the infection to its source? And how did you do it? You need to be specific about the steps. So again, make note of the steps that you're taking and the clues that you get. What were the recommendations for reducing further infections in the future? Here are some terms that are used for describing colony types, also known as colony morphologies. So just a few here. We've got elevation, margin, whole colony, and then different terms to describe those. So when you have bacteria growing on a plate, you can see them with your naked eye, okay? You don't need a microscope. But what you're seeing are not cells. These are colonies. They're large aggregations of cells. And these types of observations, these colony morphology observations, are often the first step in species identification. When you're recording the results of your hand washing and ubiquity plates, you'll want to be able to describe what may at first look like just a bunch of dots on a plate. But if you look a little closer, are those dots all the same color? How do they differ in size? Are some of them smooth and round? Maybe others are more irregular, or maybe some are filamentous. These are the types of descriptions that you're going to want to include in your lab reports. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your comments for me in the comments section below.